This audience needs no explanation of this, but blood cancer is a major health problem for Australia, and indeed around the world. And it's an increasingly common problem. Blood cancers are collectively the second most common cancer which we see in our community. And 15,000 Australians each year are diagnosed with a blood cancer, and 7,000 Australians each year will lose their lives as a consequence of blood cancer. Over 110,000 Australians are living with blood cancer, and some of you are in this room tonight. It's forecast that by 2030, blood cancer will be the leading cause of life lost from cancer. So this is a problem that we can't ignore. Blood Cancer Research Western Australia was founded in 2018. And the reason behind that was because that it became very obvious to me that the fastest way to get better treatments into West Australian patients is to offer clinical trials, and in large numbers. In 2015, when prior to starting this mission, this graph shows the number of lymphoma trials available for patients with lymphoma, which is a common type of blood cancer in Perth, compared with the two places which I'd worked in previous to this, which is Melbourne and Houston. And you can see that by international standards at that time, we were lagging substantially behind the rest of the world. I'm pleased to report that since that time, and with the support of many of you in this room, either by means of your occupation and what you do, turning up for our patients every day, or your financial contribution, the mission at Blood Cancer Research WA has resulted in a substantial increase in the number of trials which we have open, increasing from an average of 1 to 3 trials a year to around 10 to 11 trials per year over the time that this project has been in progress. And that has translated into a commensurate increase in the number of patients who have been able to participate in trials. And this is not just a number on a bar graph. This is actually someone whose life has been extended by access to a novel treatment which otherwise would not be available in Australia for years to come. And you'll hear from some of these people later this evening. So I've reflected to a couple of people in the foyer prior to starting that it's often not until I actually come to update the slide deck for evenings like tonight that I reflect on what we've actually managed to do in the last 12 months, because we're usually just too busy doing it. But it is a nice opportunity when we do this to look at the things that have happened in the last 12 months and to share them with you. So the things which I'm proud of, most proud of in the last 12 months are the continued growth of the fellowship program. And some of you in this room support the Fund a Fellow initiative, which is something which is a great idea and a brainchild of the chair of the BCRWA committee, Mark Westbrook. Thanks, Mark. And you'll get to meet one of our fellows this evening, Elizabeth Smith, who's joined us from Ireland. And one of the things which I'm sort of most proud of with this program is not so much that it is a feather in the cap for the hospital or for the state or anything else, but rather it's a direct means to allowing more patients with blood cancers to get access to trials. And we see that every day, and you'll see that when I actually chat to Lizzie on stage. But more than that, it's also a way of allowing patients with blood cancer in WA, and in fact WA itself, to become a magnet for talent. And we see some of the best and brightest people from around the world come here to train, and that is something to be celebrated. We have seen increasingly with the creation of a position for a lymphoma clinical nurse consultant, Helen Ferdas is sitting right there. We've seen the addition of incredible nursing expertise, which has made all the difference in the world to patients who are living with blood cancer. And you've heard previously from patients like Jonathan Wolfe, who's attending here tonight, at the previous meeting last year, what a difference it makes having a specialist nurse with expertise and time and compassion, who can actually listen to your story and make your journey through the health system a more smooth one. And so I can't speak enough of the gratitude that I have for the West Australian Department of Health in starting to come to the party and begin to fund these positions. We are aiming to replicate Helena's position across other subtypes of blood cancer. And as we transition Helena's funding, which initially started out as purely philanthropic sourced, it became very clear to the hospital that her position was financially self-sustainable. And they have agreed to, over a period of 2 years, fund that position, which has then become an asset to patients with blood cancer in Western Australia. And that model is something which we are going to seek to replicate across other common subtypes of blood cancer. So with a little bit of philanthropic injection at the beginning, this becomes a state asset which makes life better for patients with blood cancer, and that's what we're here for. I want to highlight an increase in homegrown trials, and I'll talk about this in more detail a bit later, but homegrown trials are a lot more work to get started than a trial which is run by a drug company. But as a consequence, there are many more opportunities which flow from these. Not only kudos for the institution, but opportunities for our fellows to participate in protocol design, which is the manual for writing the study. And these kinds of things only come about through a lot of hard work from the people involved. And finally, and potentially most excitingly, I will talk to you about the commencement of a cellular therapies program. And you'll hear from a patient of mine, Susan Leeming, who will join us on stage later, about what cellular therapies are and why they are potentially a revolutionary new treatment for patients with blood cancers around the world. So organisationally, I thought it would be helpful to show you what things looked like before we started this project in, say, 2017. The common blood cancers were there, and we had clinicians who were in charge of each of these regions. Tina Crosby, who I know is here, was the single nurse practitioner that we had, and she was responsible for everything. And that was it. That was our setup. Since the Blood Cancer Research WA project has been ongoing, we've been able to invest substantially in people who have fleshed out this structure. And now we have, as well as the lead clinician in each disease stream, we have this tier of fellows, and all of them are here tonight, actually, who have been involved in the program. Kat Lewis, Xuan Tan, and Elizabeth Smith are all here, and they've played instrumental roles in all of our success with this program. We have the fourth fellow who has been hired and is due to start in February 2022. We are in the process of creating a position for a third clinical nurse consultant. We have an additional clinical nurse consultant, Helen Ferdas, who I mentioned earlier, and Tina Crosby has now able to focus on other areas, including myeloma as well as other things. But what this has created is a really powerful platform that has dramatically increased services for patients with blood cancer, where the common subtypes of cancer are supported by 1 or 2 fellows and also a specialist nurse. And that means that patients get better care. Patients get better care means patients get better outcomes. People are living longer and they're getting much more favourable interactions with the system. All of this is really only possible with the support of people in this room. So I think I can't thank you enough for all of you for the contributions that you've made to this. I spoke earlier about delivering homegrown trials and the benefits of this. And I'll show you now a couple of trials which have been my ideas, which have now taken off. The leverage study, we're now into our 2nd year. This was an idea I had in 2016, which shows you how long it takes to do clinical research. But we were able to secure about $1.5 million from the 3 drug companies involved. This study opened around 2 years ago. We've recruited around 15 patients. You've previously heard from patients at this meeting who've participated in this study, and we will be presenting a formal analysis of this at the next toward the end of next year at our scientific meeting. But whenever I see a patient on this study, it fills me with excitement. And, virtually the vast majority of patients who we've treated have had complete responses and are free from their lymphoma. This is a common type of lymphoma, and we're getting patients into remission without ever exposing them to chemotherapy. So we're really excited about this, and really looking forward to being able to publicly share the results of this study. I also want to highlight this next study, which is due to open early next year. Catherine Lewis, who I mentioned earlier, who's spoken at this event previously, has been a fellow here going on to 2.5 years now. And because I was able to secure this funding from the drug company for this study, we were able to develop this study in Perth. This study focuses on patients with a subtype of lymphoma called primary mediastinal B-cell lymphoma. It primarily affects young women. And Kat was able to actually get really hands-on and write the protocol for this study, which is an opportunity that you really only get to do with trials that have been designed in-house. And it's being able to do these studies, which really is what helps to create a center of excellence, rather than merely being someone who just puts bums on seats, if you like. It's really getting into the nitty-gritty, and this kind of opportunity really elevates Perth on the world stage, and allows people like our fellows, and in this case, Kat Lewis, gives them the opportunity to really start to become experts on an international stage. I mentioned also the development of a chimeric antigen receptor T-cell therapy program. And some of you may have heard a little bit about CAR T-cell, as it's called in the popular media. The reason that this kind of treatment is so exciting is because it really leverages off the latest technology and 21st century technology in developing treatments for patients which are using the power of the immune system to treat their cancer rather than chemotherapy. What happens in conventional CAR T-cell therapy is that white blood cells are removed from a patient. So they have a cannula or a large line placed into their arm, and their white blood cells, their lymphocytes, which normal job is to fight viral infection, are selectively removed from the body. They are frozen in a plastic bag. They are then preserved and shipped to a manufacturing site. At that site, they undergo genetic manipulation using viruses. The viruses are designed to reprogram the cells to attack the cancer and not to damage the patient. They have specific additions of material in order to redirect them for that purpose, and also to sustain them, and they are in fact a living drug. They are then refrozen, shipped back to the patient. A patient will then receive some chemotherapy to suppress their immune system lightly, and then the cells are reinfused. These cells, when they're defrosted and infused, get busy to work. They grow up into large numbers, and they become serial killers of cancer cells. They will find a cancer cell, kill it, move on to the next one, kill it, move on to the next one, kill it, to such an extent that in a number of patients with lymphoma that is completely refractory to chemotherapy, we see response rates of 70 to 80%, including long-term cures in a proportion of patients. So this is a revolutionary treatment in patients who are able to benefit from them. And so you'll hear a bit more about that this evening. We've now opened 2 studies involving chimeric antigen receptor T-cell therapy at this site. The first one was TARMAC, which was a study led by Peter Mack, where I trained previously. And it was a study in mantle cell lymphoma using a CAR T-cell product called tisagenlecleucel, or Kymriah, in combination with ibrutinib. The second study, which is potentially even more exciting, is being done with a pharmaceutical company based in the US called CRISPR Therapeutics. And this is a phase 1 study of a drug called CTX110, which is even taking CAR T cells to the next level, because these cells are not taken from the patient who has the disease. They are taken from a completely healthy donor who has not previously had chemotherapy. Their T cells have undergone genetic manipulation with the technology called CRISPR-Cas9, or gene editing technology, which is hence the name of the company. The people who invented this technology won the Nobel Prize for Medicine. This technology allows the wielders to selectively edit the T cells to remove the part of the T cell which causes a common problem in immunotherapy called graft versus host disease. That particular molecule is edited out. And then the molecule which allows the T cell to recognize the cancer cell is inserted. So in other words, these are smart T cells. They've had the self-damage part removed, and they've had the targeting part inserted, and they get infused into the patient. There is no need for a lengthy manufacture period during which time patients can become very sick. This is a product which sits in the freezer, and when you're ready to treat a patient, you defrost the cells and you reinfuse them. And in my view, this kind of treatment is the future of cell therapy for lymphoma. And we haven't treated a patient yet, but we hopefully will do so within the next 1 to 2 months. So collectively, I think it's clear that all of these activities are putting Western Australia on the map globally and improving outcomes for West Australian patients. None of this would be possible without my incredible team, and thank you to all of you who turn up every day and do your job with such enthusiasm, and also all of our supporters, many of you whom are here tonight. Much of this, this work would not be possible without your ongoing support, and for that I'm deeply grateful.